Hi, how are you today? Inshallah fine. Today inshallah we'll start with chapter 15, selective breeding. What do you mean by selective breeding? What's the technology that it's using in the selective breeding? What's the benefit of selective breeding? That's inshallah what we will learn today. So at the end of this lesson, you will be able to identify the meaning of selective breeding, the technology that is used in the selective breeding, and the benefits of selective breeding and examples, of course, on the selective breeding. For selective breeding, you have to know that there's two technologies, which is hybridization and inbreeding technology. In the beginning of the selective breeding, hunters in the 80s needs a hunting dog that was big, strong, fast, and aggressive. All of these characteristics must be in the optimal. So they knew of two species of dogs, each with some of the desired quality, and they tried to find if they in somehow they can combine these characteristics together. So began strong but lacked speed and aggressive, this one, and this one fast, aggressive but lacked strength, and the third species big, strong, fast, and aggressive, which is the combined species between these two types. This is the meaning of hybridization. When you hybridize two species together to find a new species with a desired characteristic. Okay, then the first one mastiff, the other one bulldog, and the fourth one the bulldog mastiff. Okay. So why using selective breeding? Why we need to use the selective breeding? So as a human, we use the selective breeding, which take the advantages of naturally occurring genetic variation. We need naturally occurring genetic variation to pass wanted trait on the next generation of organisms. So we need a specific trait to be passed from one generation to another. What ways can humans selectively breed plants or animals? It's not just about the animals. We are talking also about the plants. We need a desired characteristic. We need a specific species to have a specific characteristics. What ways can humans selectively breed? Which is two types of breeding. The first one, which is the hybridization that we mentioned before. Hybridization, which is the crossing dissimilar individual to bring together the best of both worlds. So we need two individuals, and when we cross these dissimilar individual, we will take best of both worlds. The other one, which is inbreeding. Okay, the other technology is inbreeding. Let's find the characteristics of hybridization first, then we will finish the inbreeding. American Pontus Lothar Bornberg selectively bred 800 plants called varieties. Okay, and many varieties can be found in your supermarkets and bred for size, disease resistance, flowering, taste, color, and varieties of other reasons based on performance. While inbreeding, crossing breeds of individual with similar characteristics. We have two individuals with the similar characteristics, and we are breeding these two similar individuals. We are crossing breeds between these two similar characteristics. Maintains traits for or of that breed purports to find pure breeds. Okay, it's really risky because it's increased the chance for mutations since individuals are very genetically very similarly or very genetically similar to each other. So the inbreeding is more risky than the hybridization. Okay, and you will find the same characteristic that is found in both organisms, but hybridization, which is having a new characteristic that it needs already for the individuals. Let's start with the hybridization. Let's take an examples. Hybridization occurs in both plants and animals. Okay, not just in the plant, not just in the animals, for both organisms. Creating hybrid plant is different than creating hybrid animals, but you will take these two types today, inshallah. Animals can be sterile if their chromosomes number is odd. So that is really crucial here. We have to make sure that when we make a hybridization between two animals, that after the end of the hybridization, the chromosome must be an even number, not odd number. If are too many mutations that occur when they breed naturally, which is rarely occurs in the wild, okay? And plants can be bred easily by gardens or by botanists. Many crops today are hybrids, okay? Can sometimes occur naturally in the wild, sometimes in the labs. Let's start with the genetically hybrid plants. You have probably eaten many genetically hybridized or engineered plants without realizing it. Like examples, seedless grapes. This is one of the hybridized plants, okay? Watermelons, also seedless watermelon. It's another types of hybridized plant. We don't know that this is a hybridized plant, but in real, it's really hybridized because all of the grapes should be with seed. And at the same time, all of the watermelon should be in seeds. Once these grapes without a seed, that means it's really genetically engineered, okay? Bananas, which commonly have polyploidy, which is extra set of chromosomes, stronger and bigger. So the banana that's strong and long and bigger than the other, it's a polyploidy case, okay? Corn, the modern corn, was selectively bred from Tennessee at least 6,000 years ago, okay? It's already selectively bred before 6,000 years. Like this one. Look at the picture here. This is how the corn selectively breeds naturally. Look at these two pictures. Can you imagine? What's the name of these two new plants that ends from the hybridization between these two plants? Can you think about it? Okay, can you guess the hybrid fruit? This is a lime, and this is another, the common fruit. What will end with a new type? What we call it? Which is lime cutes, okay? Lime cutes, which is the hybridized species from the lime and kumquats. The other two types, this is pomelo, and this is tangerine, and the formed one, which is tangelo, which is a newly formed hybridized plant. Okay? Let's take a look on the hybridized animals. The hybridized animals are called hybrids because they are hybridized between two different species or two different animals. And when we look at these different colors, different pictures, we will find the different organisms, okay? You can find many information about the hybridized animals on the links below, okay? You will take a look at these links, inshallah, when you have this video. Designer doggies. Let's check what we mean by designer doggies. This is one of examples on the doggies. This is another one. And this is another one. Let's check. This is a hybridized doggies. Australian shaped bipedal will form Associadoodle. El Cavapu, which is Kerryville King Charles with Boodle. And this is Cheagle with Chihaho, which will form the Beagle. Okay? Look at these three different dogs. What do you mean by hybrid? The word hybrid when referring to genetics means the offspring of genetically dissimilar parents or stocks. Okay? Dissimilar parent or stocks. Okay? Especially the offspring produced by breeding plants or animals of different varieties, species, or even races. Okay? This is all about the hybridization. So again, hybridization in a plant is different than the animal, and the animal hybrids, the hybridization animals called hybrids. The plants, it's easier than the animals because in the animals, if the forming offsprings is an odd number, it is really sterile, can't reproduce. And there is many examples that you can watch on the YouTube at the link provided. Okay? Let's now check what you mean by inbreeding. Inbreeding, which is two similar genetically organisms, the best of the best. Yani when we mate or inbreed two genetically similar organisms to find the best traits. Dogs breeds are maintained by inbreedings to find the best characteristic and to keep these best characteristics. The characteristics that are unique to each breed are preserved by inbreeding. We will not bring an extra characteristics. We just keeping the desired or the unique characteristics in these organisms. Very prevalent in cattle farming. It's really prevalent. Gives farmers the best possible livestock and also can be very dangerous if two individuals have underlying mutation. If there is any mutations that will happen, it's really risky in this case in inbreeding. Okay, let's now check what you mean by biotechnology. What do you mean by biotechnology? Can you think? Can you imagine? Can you find the idea about the biotechnology? The biotechnology, which is the technology that is used in biology, and at the same time it's used in the genes to make specific characteristics. So when scientists purposely change the genetic makeup of an organism, like introducing mutation, this is biotechnology. Used in agriculture and medicine widely. The biotechnology is used in agriculture and medicines widely. Sometimes in the bacterial mutations, and a strain of bacteria has been developed that can digest oil to help clean oil spills. So we will make a mutation in the bacteria to clean up the oil spills as an example. So scientists are trying to produce bacteria to clean up a radioactive waste. This is a new technology. If it's already found, it will help us mainly all of the time. Okay, so again, biotechnology, which is when scientists purposely introduce mutation into organisms to change the genes, okay, used mainly for agricultural medicine. Sometimes, like bacteria mutation is very helpful, like the bacteria that, if it's mutated, to clean up the water spills, oil spills on the water, and they are trying, the scientists right now, to change or to mutate the bacteria to clean up the radioactive waste. Let's now check a new technology, which is combining DNA fragments. What do you mean by combining DNA fragments? What's that mean? So, since DNA is a universal code, it can be combined from two or more sources. So combining DNA, when we will take two DNA samples and combine these two DNA from two different sources, changes the genetic composition of the living organisms, and this is what we call it by recombinant DNA. So you need to know that the recombinant DNA technology, it's a specific technology that will combine two DNA from different sources. Also can be used to make proteins like human insulin, hepatitis B vaccination, clotting factors for hemophilia patients. So it's a very helpful technology. Again, let's make a quick revision for the recombinant DNA. As we remember in the last chapter, we took the recombinant DNA. What do you mean by recombinant DNA? The recombinant DNA, when we took a special DNA fragment and we insert this fragment in the original DNA. And this DNA fragment specific for a specific protein, either like the insulin hormone, like vaccination, clotting factor, and so on. Once we insert this as a new gene inside the DNA, it will be replicated and reformed again to produce these the protein or the factors or so on. Okay. Transgenic organisms. What do you mean by transgenic organism? What's the meaning of transgenic organism? Scientists find specific genes that can add them to any organism. So there are specific genes that we can add it to specific organisms. And once we add these specific genes, it will change specific protein. Look at these two cats and the picture here for specific protein that will glow in the fish here. Okay? So organisms with these genes from other species are called transgenic organisms. So we call these organisms as transgenic organisms. When we took the gene specifically from this organism and insert it in that organism, this organism will show the gene again. So we call it transgenic organism. Like red fluorescence protein, RFP in cat with the GFP, which is red under UV light, while a normal cat glow green. This is normal cat, which glow green under the UV, while this cat will glow in red color under the UV. This is a gene specifically in transgenic organism. So how they did do that, okay? Try to think, try to imagine what's the technology that is used in these organisms so they will change the glowing color under the UV. Another technology is used here, which is the cloning. Have you ever heard with a dolly goat or dolly lamb? Have you ever heard with this name? Who is dolly? What's the case about the dolly? A cloning technology or the cloning technology is a member of population of genetically identical cells reproduced from single cell. So we have a single cell that will be produced a huge organism. Dolly, which is the first lamb produced by cloning. So let's check the technology that is used here and find out what's this technology. So we will take an egg cells from adult female sheep, and we will take a donor nucleus. We will take this donor nucleus and we will fuse it with the egg cells. طبعا in the same time the nucleus of the egg cells will be removed, and in this case we have a fused cell, which is the fused cells begin dividing normally because all of the organelles that is found in the egg is found also in the donor nucleus. And after that it will form embryo, which is already foster will be added to the foster or injected in or placed in the uterus of the foster mother, okay? And then we will have a cloned organism, or here Dolly the lamb, okay? This is the first cloned lamb in the earth, okay? It's a huge technology when you take one nucleus from your cells and add it to an egg, and will form a new organism similar to you. So this lamb is similar to the donor cells, not to the mother of the egg, okay? Another technology which we make it, which is the genetic modification. What do you mean by genetic modification? What's the genetic modification? Which is a technology used to modify the plant or animal against specific criteria or specific agents, okay? Have you ever eaten a corn, potato, or soy product? Have you ever think if these products are genetically modified products or not? Try to find it out after this lecture. Many types of these products, if you will buy it from specific locations, you will find that it's either genetically modified or free of genetically modifications, okay? Many plants already are genetically modified against specific resistance to specific things, okay? Like for its already resistance to insects or herbicides or even viral infections. Many plants, if it's affected by the viral, it will dead. By genetic modification, it will be resistant against these viral infections. So genetic modification is a good process that by this technology, we can increase the production of the plants like this. To this point, we finished many ideas like hybridization, inbreeding, cloning, and genetic modification. Have a nice day and thank you.